Today, we will look into nitrogen cycle and learn what is involved in this crucial process. Nitrogen is an essential element for all living organisms, as it is a key component of amino acids, nucleic acids, and ATP. However, despite nitrogen gas making up around 78% of Earth's atmosphere, most organisms cannot use it in this form. That's because atmospheric nitrogen is extremely stable due to the triple bond between nitrogen atoms, making it biologically unavailable without conversion. Microbes drive nearly every step of this cycle. There are 5 major stages: nitrogen fixation, nitrification, assimilation, ammonification, and denitrification. The first step is nitrogen fixation. This is the biological conversion of atmospheric nitrogen gas, or N2, into ammonia, NH3. It's a form that plants can absorb and use. Rhizobium species live symbiotically in the root nodules of leguminous plants, such as peas and beans. Azotobacter is a free-living aerobic nitrogen fixer found in soil. Cyanobacteria such as Anabaena and Nostoc fix nitrogen in aquatic environments. These microbes use the enzyme nitrogenase to catalyze the reduction of N2 to NH3. This reaction is highly energy-intensive and occurs under anaerobic or microaerophilic conditions, since nitrogenase is inhibited by oxygen. Next is nitrification, which is a 2-step aerobic process that converts ammonia, or NH3, into nitrate, NO3, a more stable and bioavailable nitrogen form for plants. In the first step, ammonia is oxidized to nitrite, NO2, by Nitrosomonas species. In the second step, nitrite is further oxidized to nitrate by Nitrobacter species. These 2 genera are examples of chemoautotrophic bacteria, meaning they gain energy from inorganic compounds, in this case from the oxidation of nitrogen compounds, and use carbon dioxide as a carbon source. The third step is assimilation. This is not microbially driven, but is essential to understand the flow of nitrogen. Plants absorb nitrate, or NO3, and ammonium, or NH4+, from the soil via active transport mechanisms. Animals, in turn, obtain nitrogen by consuming plants or other animals. They digest these nitrogen-containing compounds and use the nitrogen to synthesize their own biomolecules. The fourth step is ammonification, also called mineralization. When plants, animals, or microbes die or excrete nitrogenous waste products such as urea, uric acid, or ammonia, decomposer microbes break these organic nitrogen compounds back down into ammonium, which is NH4+. Key bacterial genera involved in ammonification include Bacillus and Clostridium, which are common in soil and decaying matter. This process is crucial because it returns organic nitrogen to the inorganic pool, allowing it to be re-entered into the cycle. Finally, the 5th step is denitrification. This is an anaerobic microbial process in which nitrate, or NO3, is reduced stepwise back into gaseous forms, mainly nitrogen gas, or N2, but also nitrous oxide, or N2O, in some conditions, which are released back into the atmosphere. Common denitrifying bacteria include Pseudomonas, Paracoccus, and Thiobacillus. These bacteria use nitrate as a terminal electron acceptor in place of oxygen during anaerobic respiration. This process is especially common in waterlogged or compacted soils where oxygen availability is low. In summary, microbes play a central role in all biologically relevant transformations of nitrogen, from atmospheric nitrogen fixation to the oxidation and reduction of nitrogen compounds in the soil, to the recycling of organic matter. This concludes our lecture on this very important natural cycle. Subscribe and learn every day.